everyone, and welcome to inForm 2.4, PerkinElmer's brand new adaptive cell segmentation algorithm. What we have for you today in this video is an inForm project that we've already started. So right here we have some melanoma tissue that has been stained with 6 markers plus DAPI as our nuclear counter stain. So we have cytokeratin is acting as our tumor cell marker, and then we have a variety of immune cell markers as well. So these markers have been stained using our opal fluorophores so that we're able to spectrally unmix them and identify 7 individual signals in this same tissue section. So here we're looking at the spectrally unmixed image. And from here, I'm going to configure our cell segmentation project. Of course, in a standard project, you may want to include segmenting tissue and phenotyping and scoring your sample to actually generate some cellular-based data. But in this video, we're going to just talk about our newest feature, adaptive cell segmentation. So what's new about adaptive cell segmentation? This new algorithm allows you to reliably segment individual immune and cancer cells in densely packed, complex morphologies, including areas that have lots of inflammation. This gives you more accurate multi-parameter cell identification and spatial analysis. For those of you working with inForm 2.3, we still have our classic cell segmentation algorithm in inForm 2.4. If you're working on an inForm 2.3 project, you should go ahead and finish that up within inForm 2.3. If you want to start a new project, you're welcome to go ahead and jump in to the adaptive cell segmentation in inForm 2.4. So I'm going to configure this project with just a cell segmentation module, and we'll jump into that right now. So we're looking here again at our spectrally unmixed composite image. And my approach to adaptive cell segmentation, because this is based off of the need for a nucleus. So the default segmentation you can see across the top is nuclear. And we need to address this nuclear segmentation first before we include cytoplasm and membrane as well. So what we're going to do, we're going to go in under components, going to click on add, and when it says select a component, I can pull down the menu, and I'm going to select DAPI as my first nuclear component. So to configure, I can click on the menu to the right of the pull down menu, and I'm going to ask inForm to use DAPI to find our nuclei. Now, those of you who have worked with previous versions of inForm in the past may notice right away our obvious new feature, which is this live preview window. This live preview allows you to see in real time any changes that you make to the segmentation or to splitting. You can see the effects immediately without having to segment the entire image. We can move the live window around the image so we can look at different cell types, and we can zoom in to get a closer look at exactly how our nuclei are being segmented. When I am looking at nuclei, one thing that I find easy is to look at just the DAPI composite image. So here now, instead of looking at all 7 colors at the same time, I'm looking at a pseudo H&E image of just the nuclear signal. And that helps me make my first decision. When we're configuring DAPI, we have a slider bar to adjust the typical intensity. And so what this does is it allows us to determine how much nuclear signal is being collected. At this point, we're not segmenting yet. We're just making sure that we're picking up enough nuclear signal. So you can see as we slide the bar to the right, it's a little bit more sensitive. We're picking up less signal. As I slide the bar to the left, it's a lot more inclusive. We're picking up a lot more signal. Again, at this point, we're not worried about the splitting. We just want to make sure that we're picking up all of our signal. So here I'll settle at a value pretty close to the middle. Looks like I'm getting a lot of my nuclei somewhere around here. So once I'm satisfied, I can click OK, and we can always go back and adjust that value if we want to a little bit later on. Now that we've picked up all of our nuclei, we can always double check by turning off and on our nuclear mask. So if I click on this icon, I can turn off and on the results and make sure that I'm picking up all of my DAPI signal. Once I am satisfied with that I'm picking up the signal and that my relative intensity is at a proper value, now I can scroll down and start looking at my nuclear splitting. So when we start splitting, we have another configuration window. And if I click on the configuration window, there's a few parameters that I can adjust that'll help me with the splitting. So based on the quality of my staining, there's some preset options that can help get me started with my splitting. So is my tissue, are my nuclei unevenly stained? Are there some holes in the center of the nuclei? Is there a mixture? Are some nuclei nice and punctate? Are some of them with holes? Or are they all evenly stained with very few holes? If you pick the right option, this can give you a head start with the splitting. I typically end up most of the time settling on a mixture of quality. Then I can adjust the spider bar to aggressively or less aggressively split my nuclei into multiple cells. So I have my eye over here, want to make sure that these two guys are split into two separate cells. If I turn this mask off and on, I can see I'm getting really nice splitting of those multiple cells. Again, I can move the window around and check to make sure that now my splitting is accurate. So now that I've worked with the DAPI signal, we can go back up and we can add other compartments or other components to help us segment the nucleus. Well, one thing that I want to point out is if we want, we could also add an additional nuclear marker. So in previous tissues that I've worked with and other staining, sometimes when I have a nuclear marker such as FOXP3, it can kind of overwhelm the DAPI signal. And if I'm only using DAPI to detect my nuclei, some strongly FOXP3 positive cells may not be counted as nuclei. So inForm 2.4, the adaptive cell segmentation, takes that into account, and it lets us add additional nuclear markers to help find our nuclei. One additional component that I would like to adjust before we start adding different cellular components to it is this minimum nuclear size. And so we can adjust how small nuclei are so we don't end up with very tiny over-segmented cells. Now, this really will depend on your cell type, your tissue types, what you're looking at, but I tend to like to leave it around 60, maybe we'll drop it down to 50, just so you can see an idea of what that looks like when we change it. If I scroll back up, now we're happy with our nuclear segmentation. We can now go in and we can add different cellular compartments to help aid our nuclear splitting. So we can use cytoplasmic markers or membrane markers to help our splitting. So in this case, we're going to go ahead and we're going to use CD8 as a membrane marker to help split our immune cells. It can help to have a pan-immune marker if you want to help split the nuclei of your tumor cells because they're typically much larger than your immune cells. It could be useful to use cytokeratin in this step to help split nuclei of different sizes. So I'm going to go ahead and I'm going to ask inForm to use my CD8 signal in the membrane to assist splitting our immune cells. All right, click OK there. We have a few more options that have popped up. We now have an option of assisting component splitting. So this is referring to cells that are CD8 positive. Do I want those to have a different minimum nuclear size than other cells in our sample? So if I know my immune cells are typically going to be a little bit smaller, if my cytotoxic T cells are a little bit smaller than my tumor cells, for example, maybe I would want my minimum nuclear size for my assisting component to be a little bit smaller than my minimum nuclear size for my nuclear component. If I click on this configuration menu, then we have a few options, again, correlated to the quality of your staining. So is your membrane staining, is it a little not specific? Is it a little discontinuous? Or is it very precise and continuous with very sharp edges? Or, as is most often the case, is it a mixture of quality here? So that's what I'm going to select. Again, this is very dependent on your sample and what's working best for you and your specific tissue. So again, I can adjust the slider. I'm going to zoom in a little bit so we can see results. And here again, if we move around, I'm actually going to switch to see some CD8 positive cells. And we can see now adding in the CD8 component can help us split our nuclei a little bit more accurately. So with that, we can go ahead and continue to add as many components as we like. Again, it could be useful to include cytokeratin to help segment your tumor cells. For this example, I'm going to just leave it with DAPI and CD8. Once I'm happy with the nuclear segmentation, then we can go in and add cytoplasm and membrane components as well. So here, for the nuclear segmentation, we added a membrane marker to help us segment the nuclei. Now we're actually going to add a little bit of cytoplasm and membrane search distance information to help segment the cell fully. And so we can actually get some quantitative data about these markers downstream. So as we turned on our cytoplasm and our membrane components, now we have options to adjust the thickness of the cytoplasm and the search distance from the border of the nucleus out to the membrane. And again, these are very tissue specific. It's going to depend on your sample and your staining. We tend to like to adjust the defaults a little bit. For cytoplasm, we like to increase that from 3 to 6 pixels. And for the membrane search distance, we like to adjust that up a little bit. We found that 20 pixels tends to work pretty well for us with our sample. So now as we go to segment, we can see a nice nuclear segmentation. We have our cytoplasmic segmentation, and we've got our membrane segmentation to finish it off. So once I'm happy with what I see in my live preview window, I can move back to my composite multicolored image and segment the entire image to see how my whole cell segmentation works across the entire image. Now, once the cell segmentation is complete, it'll generate a data table here where we can actually start quantifying our sample. And of course, from here, then you can move into phenotyping your cells and scoring to set thresholds and get positivity for 1 or 2 multiple markers per cell. So I hope this has been helpful. Good luck with your analysis projects and enjoy cell segmentation.